like analyses I'm doing. I think currently it'll be more one of the data sets. Oh, why
Right. Okay. What do you want? Cool. Morning, everyone. So today we have guests from University of Leeds, Emma, James, and Danai. So guys, we've been doing some work in the Kruger, which is quite an amazing trip that I had with them. So guys are working with Josie South, who's an associate professor at University of Leeds. So I think the plan for today is mainly, we have 3 different talks, but the guys, they want to have 10 minutes each, and then questions after 5 minutes. And then at the end, maybe have a discussion and see how things go. So first up is Danai. So Danai is a postdoc, has a background in conservation ecology, population genetics. Currently, she's looking at the traits of crayfish, which is a huge problem. So I think what these guys are offering to us today is quite extremely important. And also mainly understanding the impact of invasive species, which is quite important, and some of the work that SAIAB is actually very much involved in with Professor Julie Coetzee. So over to you, Danai. Yeah, looking forward to the talk.
Thank you. It's the control for the slides. Can I just do it from here? Oh, okay, perfect. Okay, thank you so much, Liba, for the introduction. I'm really happy to be here, and thank you to SAIAB for hosting us to do a part of this work that you'll see here. So this is a work that started back in, well, quite a few years actually, but my talk will be focusing on the work that we've been doing in Kruger National Park over the last 2 years. So most of the data will be from these last 2 surveys. So you will hear a lot more about the work that we're doing in the entirety of the community from James and Emma. My job is to give you a brief introduction about the crayfish themselves and how we survey and what we've seen. So the star of the talk is the Redclaw crayfish. It is originally from Australia and Papua New Guinea. It is invasive in South Africa and other places in Africa. The main problem with crayfish is that they're omnivores and they can insert themselves in all levels of the trophic food webs. They're highly fertile. They've got a lot of traits that make them ideal invaders. And they are a very popular aquaculture species, this guy in particular in Australia, and this is the main pathway with which they've been introduced, mainly as aquaculture species or as pets. So what's the problem? As I said, they interact, they compete, they feed with many different organisms across the aquatic trophic food webs. And this can create a series of problems, mainly altering their habitat, so creating structural damage in riverbanks, especially for burrowing species, transmitting novel pathogen, and Redclaw in particular, and Josie has done a lot of work on this, is notorious for damaging fisheries. So they will eat the fish, they will damage equipment, and that results to huge monetary losses and great socioeconomic impacts for the communities that depend on those fisheries. So this is a map of Kruger National Park, so we know kind of, so just to give you a bit of a spatial context, so this was originally presented in the Savannah Science Meeting at Skukuza. You're definitely not here, but when the original, yes. So, but it's still a bit useful just to give you again a bit of spatial context. So this is Skukuza, the main camp of Kruger National Park in the lower parts of the Kruger, and this was our hub throughout those surveys as well. So where are the crayfish? This was the original question. There were reports that they were running around Kruger National Park, but we had no idea where exactly they've been introduced. So Josie led a huge survey in 2024 where the team surveyed the entirety of Kruger National Park, so from the top to the bottom, all of the rivers, just to see where crayfish are even present. Thankfully, at the time, they were only found in the lower Kruger, in the Inkomati River Basin, in the Sabi Sand and Crocodile rivers. So in 2025, last year, we set out again to sample only the lower parts where crayfish were present to see how are the populations doing, are we catching more, are we catching less, and also see if there are any changes in those populations and if we can get a little bit more information on how they're behaving and how they're impacting communities. So a bit of a timeline of introduction based on what we know. The first records of Redclaw crayfish in Kruger National Park were from 2016 in the Van Haren Dam at the very bottom of the park in the Crocodile River. And since then, we know that they have spread both upstream and downstream of the Crocodile River. For the Sabi Sand, we believe that the introduction was around 2022, just outside of the park in Exeter Weir, which is part of the Sabi Sand Reserve, which is one of the conservancies that are directly attached around the borders of Kruger National Park. And we have since then seen a steady progression of the crayfish downstream. So these are all of our main sites that we've been monitoring for the last 2 years. And another important term that I would like you to know of before I move on, just because I mentioned it in my graphs, and you will hear more about it from Emma and James as well, is what we refer to as the invasion gradient. So for the park, we've got our core sites. The invasion core is basically the first site where we got a record. It's what we consider to be our introduction site. Then we've got our intermediate sites. Those are the sites that are immediately upstream or downstream within, I think our calculations were about 50 kilometers from the original introduction site. And then we've got our front sites, which is basically the furthest point from the introduction site downstream where we have recorded crayfish. So how do we actually record the crayfish in the field? It's actually a pretty simple process, though I feel like I shouldn't call it simple because it does take a lot of effort, especially when you're chased by crocodiles and hippos in Kruger National Park. So thanks to our lovely guards who made sure we're safe, we find good spots that would be suitable habitat for crayfish. They usually like overhanging vegetation, places where they can go and hide in the rivers. We set out traps in the evening. They're a very easy, a very simple device. We put baits, crayfish go in overnight, and then we lift it out in the morning and see what we caught. So important measurements for that are the catch per unit effort, very similar to what is recorded for fish. It's our way to standardize abundance and compare between sites, and the detection probability, which is how many crayfish we caught versus the traps that we actually deployed. So once we actually have a crayfish at hand, the first thing we check is its sex. The mass, I promise you we use scientifically appropriate scales in the field. Then we take some key morphology measurements that are related to their behavior. So the claw lengths, the front leg lengths, and the carapace. So we have an idea of how big the animal is and how that what that means about its movement and its interaction with other organisms, as well as competition between them, the crayfish themselves. And the other thing that we've been doing for the last 2 years is collect tissue samples. That's mainly muscle and gut. And this is what I'm actually doing here in SAIAB with Emma's help. We're processing those samples for DNA and gut microbiome. And we hope to have some more exciting information about what their populations are doing, how they're adapting, and how their microbiome changes along the invasion gradient, as well as compare different populations across South Africa, because population genetics is an amazing tool and it can actually tell us, help us trace introduction pathways as well as movements. So My first question that I started with is, where are the crayfish? So this is Lower Kruger, and this is what it looked like in 2024 in terms of crayfish. So these are all the sites that were sampled. The X's mean places where we sampled but did not catch any crayfish. And this is what things looked like in 2025. So you can see that we have some new records in Lower Sabi, and we've also in 2025 surveyed Exeter Weir and Malamala, which are in the Sabi Sand Reserve, and we found some crayfish there too. Another interesting result that was almost an accident when I was comparing data and going through is the difference between two methods. So trapping is our standard way of catching the crayfish, as I mentioned, but at the same time as doing the crayfish, there were lots of surveys going on for fish, for invertebrates, for isotopes. So people were generally sampling. And while people were electrofishing for the fish monitoring, we also caught a lot of crayfish that way. We recorded them and kept those data aside, but I was interested because I noticed that there were some differences between the two sets of crayfish. So I looked into it. And in some cases, you can see that basically we caught more crayfish electrofishing than actually in the traps. And the other important thing, which could be useful from a management perspective, is that we saw a bias between the two methods when it comes to crayfish size. So we're missing out a lot of the smaller, younger crayfish with the traps. We're actually catching them electrofishing. The other important bias is that we seem to be missing a lot of females from the trap as well. We did know that there was a slight bias towards males just because we put bait in the traps and they're more competitive and tend to be more aggressive. But again, from a management perspective, if you want to remove reproducing females, it might be best to actually combine the methods based on the data that we've collected from the last 2 years. And we're seeing something similar in the UK as well, where we're also monitoring different species of invasive crayfish. So just to give you an idea of the spread rate, I've already showed you kind of where they are on the map. But for the Crocodile River, we estimate that they have a spread rate of about 4.5 kilometers a year. For the Sabi Sand, that changes to almost 23 kilometers a year. And if we take into account the difference just between 2024 and 2025, we saw a 35-kilometer spread downstream after heavy rains in 2024 from Antlister to the Lower Sabi, which is quite scary, to be honest, especially since Kruger had major flooding again this year. So I would personally be quite terrified to look at what crayfish are this year, probably absolutely everywhere at this point. This is the difference in catch per unit effort between the 2 years, 2024 and 2025. You can already see that we have a lot more crayfish in 2025 for a lot of the sites. And just to give you a number, for the Crocodile River, this is like a 3 to 5 times increase in the catch per unit effort and the abundance of crayfish just within a year. And this is true and consistent across all sites. Like we are seeing an increase in the detection of crayfish over the years since introduction across the Kruger National Park. So another thing that I was interested in were how the crayfish populations are changing. This is based on morphology. This is the difference in mass between 2024 and 2025. And this is the difference in carapace length between 2024 and 2025. Now you can see the purple bits at the very end. That's the comparison for Kruger National Park. The gray bars represent other invasion cores from previous years. And you can see how big the differences within the year for Kruger National Park. And you can see that the sizes that we're seeing now in the Kruger are pretty much double from other introduction sites as well. So it's a huge, huge difference. And we're now interested in seeing what the genetics will tell us and how this is going to develop over the next couple of years and what it means for their impact. And this is particularly prominent in the earlier parts of the invasion core, so in the core and intermediate sites, and less obvious at the front. So I looked into this a little bit further, trying to understand what is driving the spread of crayfish and why we're seeing these differences in size. So this is an aggregated data set that includes both the purple and some of the gray bars that you saw earlier. So this is data from the 2 years at Kruger National Park and from the Barotse floodplains. This is just a PCA, a principal component analysis. I was trying to get the measurements of the crayfish to check my individuals and see if splitting them by site, by river, by year was going to give me some sort of split. Nothing seemed to matter except for the morphology data along the invasion gradient. Is not as prominent, but we are basically seeing that in the earlier stages of the invasion gradient, so in the core and intermediate sites, the individual measurements, so the legs, the claw lengths, and the carapace seem to be playing a bigger role, which suggests that potentially competition is playing a role as to who gets to progress to the next stage, let's say. But in the front of the invasion, we see the bigger crayfish, the bigger overall crayfish leading the invasion. The GM over there just means geometric mean. It's basically an aggregated measurement of all the morphology with some corrections. I'm not going to bore you with the details, but that's basically what it means. It takes into account the full morphology of an individual. And the other two important parameters were the distance from the introduction site, which was significant. Basically, the further away you are from the introduction site, the less crayfish was catching, which we kind of expected. And then the other important factor, why I mentioned the rainfall and the spread rate earlier, is that another significant driver of the crayfish abundance was the average rainfall of the previous quarter. So the more rain and water, the more crayfish you've got, which makes sense. However, it is alarming given how the climate is changing and how much the rain is intensifying last year. So overall, we once again say that crayfish are a huge threat for natural protected areas, especially because in Kruger, but also in South Africa overall, there are no native crayfish whatsoever. The only native analog is a crab. So we don't know how they're gonna interact with the communities. Well, we're learning. You'll hear more about it from Emma and James in a second. But just some key points to summarize before I go. We've seen rapid spread and establishment within Kruger in just the last 2 years. We believe that extreme weather might facilitate further spread. And we've also have some evidence that competition and body size is increasing along the invasion gradient. So overall, there is an urgent need to monitoring and assess their impacts and to manage the crayfish. And we're doing this work for both the Redclaw crayfish, but also the red swamp crayfish that's been introduced. I'm currently processing genetic samples for both, and we're trying to understand where they're coming from and how they're behaving. And the main thing that you can do as well is just raise the alarm, be aware. And it is very much a work in progress, and SAIAB has been playing a huge role. So thank you for being here, and thanks to absolutely everyone who made it work, you were included, and other people who are mentioned here in the photos that just made it possible, because it was a huge effort in the field to get this data out there. So that's me, I think. Thank you very much.
All right. Cool, nice. We have questions for Danai. It's really interesting. I have one. So it's a good thing you mentioned this is quite worrying and scary, but how long should we wait? And also, what interventions would you suggest as early as possible and how to manage this? Kruger is massive in terms of manpower. You need a lot of money and resources.
We're saying the same.
Yeah, but we need something.
Yeah.
We need something urgent. What would you suggest based on your expertise? Yeah.
That's a long, big discussion, basically. I think there is some work being done. Josie is very much pushing this for, like, basically giving the equipment to people. Anyone who's monitoring freshwater system, essentially, what we've been trying to achieve is anyone who is going out in the field to collect fish, because they're doing all the work already, dropping nets, it's very easy to drop a few traps and keep an eye out for the crayfish. We also spend a week trying to talk to people around Port Owen about potential introductions of red swamp crayfish. So at this stage, I think raising awareness and keep monitoring, which is expensive and difficult, but if it can be done alongside other surveys, then I think that will be the best way forward.
And one of our colleagues is modelling different management strategies to see which are the most effective, like random trapping versus that target trapping. So there are steps there's research essentially being done into the best way to manage it. But essentially, we shouldn't be waiting.
We should. That's the main thing. We are working on an updated protocol with Josie as well, and we're hoping to incorporate the data that I just showed you with the difference between the methods to hopefully have a bigger sort of standardized protocol that can help.
Oh, and then Francesca has a question.
Yes, please. Thank you so much, Danai. Very, very clear presentation. I've got one comment and another comment for what's next, which I don't know if any of you is already covering. The comment is when you show there was very much overlap in the front, intermediate, and the core, and then you explain it based that the biggest are the ones that are mostly moving and driving this extended invasion. Wouldn't that be natural that those larger individuals somehow were skewing the data set and pulling the data set in that direction, because it was very much a cluster with some of outliers, if you want to call it.
Sorry.
Yeah.
So I based that, I'm a bit more confident just because I've seen that pattern in every single introduction call where they've done 2 years repetition, and we're seeing something sort of similar in the UK as well. Very, very early data sets, which is why I would take this with a pinch of salt, basically. So I think this is what's going on, but it is still quite early, and I'm hoping to have more data. However, I am working on a massive global crayfish trait database that includes a lot of records from crayfish from the same species in both their native and invasive range. So I am hoping that this year I can answer that on a much larger scale. So stay tuned with that, because it's a very good point.
And the next one, sorry, is just that as a larval ecologist, the next step, have you tried to understand the connectivity of the population connectivity from an early stage population? So where are these larvae and where are they coming from and where are they going? Especially with these beautiful brains that basically connect everything.
Yeah, so the crayfish tend to keep all of their young on them. So I think the main, correct me if I'm wrong, Emma, James, I seem to remember that the main way is that the female will literally just carry them on. Yeah.
But they also can.
They can.
The young of the year are tiny, and they'll get on like rapid vegetation and those.
I'm not aware of any.
I haven't looked into it specifically.
Yeah, I'm not aware of any survey that specifically looked into that.
Because that would be the quickest way of being transporting as a, I know
I
that
agree with that.
They're brooders, but they still release something.
I agree with that, yeah. I think that's the main reason why we're seeing them pop up rapidly after the rains, like they're being flashed down. So, and the little ones are very little, so difficult to spot.
Can we ask you afterwards how you model that yourself, or how you that? Maybe it's
Light
like
traps.
now.
Light traps, yeah.
Vera has a question.
Well, thanks for the amazing talk. This is coming from a person that is not familiar with the word. Can you truly say that there was no Redclaw fish crab in a system? Like, can you be confident to say there was not, or was it not detected? But how confident were you?
Not as much as I would like personally, because we base this based on like it hasn't been seen, it hasn't been reported, but obviously with any species, unless someone is there 24 hours, 24/7 trapping and staring, it is always a challenge. I think that's true for any kind of monitoring, especially if you've got something that's at a lower density. And with these, unfortunately, by the time you hear about it, it's when it's increased in density and people start noticing it, and they will tell you, hey, this thing is appearing in the river. So it is difficult. It's one of the main challenges in invasion ecology as well. And I'm hoping that the population genetics will give us some idea of where they might be coming from and when, potentially.
Thanks for your presentation. Following Buyo's question, have you tried other methods like the eDNA to detect crayfishes in the system?
We haven't, but I'm pretty sure there are markers for it. I am advocating for it at the moment. This is the first, at least for a lab, that we're doing the genetics for it. Right now we're doing the actual samples, but I'm very much advocating that we add eDNA to the mix.
Someone in our lab did it in the UK, and it didn't work.
It didn't work in the UK. They don't shed as much though.
Unless you've got like a whole mole. then they're actually quite like it relies on them shedding stuff. So he was sort of like looking in a pool with crayfish in, taking the water out and then
It was coming blank, yeah.
So
It is a useful monitoring method, but yes, James, you're right. I forgot that they tested that for the.
So you mentioned that you're collecting samples for living
Mhm.
CCTV. Have you just collected the samples or have you started the extraction process?
We're in the process of extracting them up still. Right now, we're just using a commercial kit for DNA extraction, but it's just muscle tissue, so can be done with a number of protocols, really. This one is just we happen to have tested it before. It's the QIAGEN kit, the blood and tissue kit, and it works well for it. But there are other cheaper methods as well.
One more.
Okay.
Is there any place in the world where an invasive crayfish species from any lineage has been controlled while free living in lakes or rivers?
I'm not entirely sure if I can answer this question because I don't know. We might have to check that. I think there are some.
A lot of time they're at such a high density that you cannot. That's why we're advocating for the proactive management, because if you get them while they're at low abundance, then you can maybe initiate like population collapse, and that can help you finish the job.
They're also
there
very
are-
resistant.
Yeah,
We've heard some crazy stories while we've been here trying to investigate essentially things popping up and us not knowing how they got there and people being like, oh yeah, there was a farm here like 20 years ago.
And they've been eaten.
no,
Yeah, but I wouldn't recommend it. Now some of them from some places where we fish them out of, I wouldn't eat them. But yes, they are very much edible. They're actually quite popular, apparently very tasty.
but we should not be going down that line.
Yes.
That just once there's value associated with them, then
We don't want that.
even more. With the eradication, I think it's been successful in like lakes and things which are isolated and not interconnected. You can be a lot more sure that you've got rid of something. With the Kruger, it's more like sorry, go for it.
I think it doesn't. It matters less if they're completely gone. It's more we don't sort of detectable impact of them. So that's what we're.
Maintaining low densities in
Yeah.
areas that they're already there and stopping them from getting into areas that they're not there yet.
Because it's difficult.
Rivers are also incredibly challenging, especially when you have places like the Kruger, when you've got rivers flowing in and out from 3 different countries and like 2-3 different basins. So we try, but the main thing is to not get them in the system, which we always try to just don't get them in the system to begin with.
And then once they're in, keeping low densities and keeping them from going elsewhere.
Sorry, I mentioned about the eating because in the Mediterranean, the invasive crabs has got so wild, literally, that there's no other control than trying to persuade people to favor one source of food versus the other one. I'm not talking about aquaculture, but
Yeah.
mechanically, that's how they, and I'm not an invasive biology specialist.
They're not very popular though. Josie has done some work on the perception of the
Okay.
crayfish with the communities on whether it can be sustainable to fish them out of the system and actually use them as food, but people don't really like them because it's a novel thing and they'd much rather have, well, the fish that they know.
I was going to say, I suppose, like, if we get to the point where they're that abundant, then sure, may as well give it a try. But at the moment, we want to steer away from that kind of thinking. You can lead to informal agriculture, which is like probably where they came from in the first place. In the UK as well, they tried to eat our invasive crayfish and it hasn't worked.
I know these guys will go on for 30 minutes. James, wait. Okay, no, just one more. Just one more. There's one online from Kebuko. Just one.
Yeah.
James?
Hi, thank you so much for sharing all this information with us. I'm just having connectivity issues here and I might have missed this. There's a slide where you were talking about different catching methods that were catching different sexes or the abundances were different.
Yep.
So I'm just curious to know what could be the reason for the sexes, the abundances on sexes being different based on the catch method.
We think it is a case of behavior. I I haven't formally studied it. I'm just presenting it as a part of the study, but we have seen it in the UK as well, that males tend to be more aggressive and more competitive for the bait. So we end up catching more of them in the traps. I hadn't realized how much of a bias it was until I started comparing the Kruger data.
Okay, and was it more females through electrofishing?
Yes, yes. So we caught more females. The other
Do you
idea
think
that
that could be the same for fish?
I am by no means a fish expert, and I am not making speculation in front of a room with fish experts. I'm quite new to crayfish myself, but I especially with fish, I will not dare. Might be interesting to know, but
We didn't sex them, so.
Okay, cool. Thanks, Deboko. And then you're done. I think there's some.
we have the samples.
Yeah. So next speaker is Emma. So Emma, she's a PhD candidate. Also looking at crayfish, effects of crayfish, but what's really interesting is the aspects on macroinvertebrates. I think that would really be nice. So over to you, Emma.
Thank you, Luba, and thank you everyone for being here. It's a pleasure to be at SAIAB. So yeah, as Luba said, today I'm going to be talking, we've heard about where crayfish are in the Kruger, but now we're shifting gear and looking at how the macroinvertebrate communities of Kruger are responding to crayfish invasion. Wait, how do I make this move?
It's the arrows, Emma.
Okay, cool. So before I go into it, I just want to tell you guys a bit about what I'm about more generally in terms of what my PhD is about. So this is my PhD title, and I have like kind of 4 core research questions that I'm seeking to answer with my project. So how do crayfish invasion impacts evolve across time and space? So looking at context dependencies, things like environmental factors, time since invasion, how these things affect the magnitude of impact that we observe. And then what I'm going to be talking about today is how macroinvertebrate communities respond to crayfish invasion, and then speaking back to the context dependence of the magnitude and such. And then another part of the Kruger project that we're working on is how crayfish integrate into aquatic food webs, so using stable isotope ecology to try and answer that question. And another thing I'm working on is ecomorphology and looking at crayfish morphological traits and whether these things can predict impact as well. So today we're going to be hearing about the macroinvertebrate impacts. So macroinvertebrates, just in case anyone doesn't know what they are as a term, they're all the bugs and beasties that live in your rivers, things like your gammarus, so shrimps, snails, mayflies, stoneflies, things like that. And these things are the foundation of aquatic food webs. A lot of things eat them, and a lot of things rely on them for things like nutrient processing in river systems. And the composition of macroinvertebrate communities can indicate overall river health. There's lots of metrics associated with assigning taxa certain values based on their sensitivity to things like pollution and such. So they're really good biomonitoring tools. And as such, in South Africa, we have a tool called the South African Scoring System, or SAS. I'm sure many of you are familiar with it. And this is a rapid assessment protocol used to look at macroinvertebrate compositions and see how they're responding to anthropogenic stresses. And fortunately, Kruger National Park started doing SAS monitoring in 2010. So now we have a lovely long-term data set with community composition, pre and post crayfish being introduced in the park. So now we can see how crayfish, how macroinvertebrates are responding to crayfish. And this, as you've already heard, is our crayfish in question, the Redclaw crayfish. And they're potentially very detrimental invaders for a number of reasons, but from a macroinvertebrate perspective, they're competitors with macroinvertebrates, they can predate macroinvertebrates, and they can also alter their environment and habitat. And as we heard from Danai, the first record of Redclaw crayfish was in the Crocodile River in Kruger National Park, and this was in 2016. And since then, we've found them on the Sabi and Sand rivers in 2022. So this project, we have this lovely long-term monitoring data, so there's a billion questions we could ask with it. But today I'm going to be focusing on the macroinvertebrate diversity, identifying hotspots of diversity across Kruger National Park, determining how macroinvertebrate communities are responding to crayfish, and identify which macroinvertebrate types are being impacted by crayfish, maybe disproportionately. So first, let's zoom in on the Kruger and have a look at how the diversity is different across the park. So here's a map of Kruger National Park, and the circles represent biomonitoring sites from across the park, and the colour represents the average richness of those sites. So the darker colours are higher in richness, the lighter colours are lower in richness. And just to zoom in on some individual rivers, this is the Lavuvu River at the top of the park. And now the circles are different sizes to represent the sampling effort, so how many years that site was sampled for in the biomonitoring data set. And we can see that the Lavuvu generally has quite high richness, number of anthropogenic stressors acting on this river system. And then moving down, we have the Letaba and Oliphant rivers. Again, pretty good richness, a lot of anthropogenic stressors acting on these rivers as well. And now we have our SAIAB River at the top and the Crocodile Rivers at the bottom. And these generally have really quite high richness as well. But I would say that the SAIAB is a particular hotspot for macroinvertebrate diversity in the park. And importantly, as you heard from Danai, the crayfish introduced onto the Crocodile River 2016 and on the SAIAB River in 2022. So there's a lot of macroinvertebrates in the park, but how are they responding to crayfish? So I had to come up with sort of an experimental setup using the rivers in the park to look at this. So we have the Crocodile River. We know that was invaded in 2016. So I've treated anything pre-2016 as uninvaded and anything post-2016 as invaded. And likewise for the SAIAB and Sand rivers, anything pre-2022 is uninvaded and anything post-2022 is invaded. But we want to be sure that any shifts that we do see are driven by crayfish and not just other environmental factors that might drive community shifts, because macroinvertebrates are responding to more than just crayfish. So to try and tackle this, I have a control system using the Letaba and Oliphant rivers. We currently have no records of crayfish at those rivers in Kruger National Park. And I've taken an average introduction date of crayfish in Kruger National Park, which falls at 2019. And this is sort of like a baseline for when communities might be shifting. So for these control rivers, anything pre-2019 is pre-crayfish as an average in Kruger, and post is post-crayfish. So here's some results from my control site. So this plot is a bit confusing, but I'll just walk you through it. So each point on this plot represents different invertebrate communities and at each of their corresponding years. And the lines connecting them shows you kind of like the trajectory of the communities and how they're moving. And the coloured polygons surrounding them, the green represents pre-crayfish as an average, and the orange represents post-crayfish as an average. And the general take-home message from this plot is that there's still quite a lot of overlap between pre-crayfish and post-crayfish communities, and there was no significant difference between them when I tested this formally. And then to do some more examples of different sites on the rivers at the top there, we see that as a pattern across multiple sites across the park. And I should say that I chose the sites with the best temporal spread of data to try and capture those differences pre and post crayfish. And again, none of these came up as significantly different. So it seems like communities aren't really shifting significantly in response to other environmental factors. But how does this change when we add crayfish into the mix? So the answer is quite a lot, actually. So colours have changed now. So this is a site that we know has crayfish, and the blue is uninvaded and the pink is invaded. And we can see that they've kind of shifted quite a bit, and there's not very much overlap between these two polygons now. And when I tested this, this was a significant difference. And then I wanted to know who was driving these shifts, or which macroinvertebrate taxa are maybe driving responses. And the short answer is there's lots of small contributions by lots of taxa. So that kind of indicates to me that there aren't any particular taxa being lost or gained. It's more of a maybe fundamental change in the overall composition. But in a slightly different analysis, I did find that these two taxa, so elmid beetles and leptophlebid mayflies, were significantly associated more with uninvaded communities, and not really sure why this is. So if anyone has any inputs on that, that would be awesome. But my working hypothesis is that they're both scraping taxa. They like sort of scrape algae off rocks, and maybe crayfish are either competing for them with them for that resource, or crayfish just moving around is sort of like disturbing that habitat for them. But yeah, if anyone has any thoughts on that, they would be gratefully received. And this is a different site on the Crocodile River this time. And again, we see this quite large shift between uninvaded and invaded communities. And again, this is a significant difference. And who's driving these shifts again? This time, it was not really anyone in particular. So again, sort of like a fundamental difference in community composition. And what I'm working on now is whether we can use these shifts to sort of predict when crayfish might have entered a site, because that's a really big question in invasion ecology, is you can never really narrow down when something actually entered a site. So here's two sites that are on invaded rivers, but we haven't had any crayfish records on them from our 2024 and 2025 sampling. And we can see that Sekirawani on the Sabi and Sand rivers is experiencing quite a big shift. So maybe crayfish were around that 2022 mark. But yeah, TBC. And again, on the Crocodile River, we see that there's not really much, there's a lot of overlap, so there's not really much shifting. And this site is like really far down on the Crocodile, basically almost outside of the park. So I think that they probably haven't got that far yet. And hopefully it will stay that way. So my take-home messages for all of this work is that Kruger National Park has a lot of macroinvertebrates, and we want to protect that. And the Sabi River is a particularly diverse place, which is of concern because we know that crayfish are there as well. Also, long-term biomonitoring is a really vital tool to assess how aquatic communities are responding to environmental stresses. I've talked about invasive species, but it could be anything. So yeah, if we didn't have that long-term data, then we wouldn't know what the communities were like before and then after. And I've showed some evidence of macroinvertebrate community shifts in response to crayfish invasion. But macroinvertebrate communities can recover from things like droughts and flooding, so hopefully the same is true for invasions. So future work on this, because this is very much the early stages of me getting into the data set. I want to do a functional diversity assessment, which is basically assigning traits to all of the macroinvertebrates that I have to see if there's specific traits that are being disproportionately affected. So maybe leaf shredders are being more effective, maybe softer taxa are being more effective, and that can help us predict who might be impacted by crayfish invasion in future, and also help us predict consequences for ecosystem services that macroinvertebrates provide. And also of potential interest to anyone who does do SAS monitoring themselves, I'm working with a team of people who made an R package called BioMonitor, which is used for European biomonitoring data like SAS data, and we're trying to implement SAS into this package so that you can have easily automated and reproducible workflows. And we're trying to integrate it with trait databases for South African macroinverts as well, so you can do functional stuff as well as your taxonomic stuff. So just want to say a huge thank you to everyone who collected the long-term data. I was lucky enough to come to Kruger last year and I had the best time. And yeah, thank you so much for listening, and yeah.
Cool. Questions open. Okay, there's one from.
Yeah.
Hi, Emma. Thank you for the presentation. Interesting I assume it's probably, I'm not a crayfish expert, so.
It's okay.
Assumed knowledge, but what exactly is the trophic ecology? What do they eat? And beyond what do they eat, what do they actually eat right now? Because maybe having that knowledge can actually help you understand and interpret the data that you have.
So the answer to what do they eat is everything, which is why we're quite worried about them as an invasive, because they'll eat the inverts, they'll eat the plants, they'll eat each other. So yeah, their trophic ecology is quite complicated, and a particular interest is the fact that they're in Africa, because Africa as a whole continent doesn't have any native crayfish except for in Madagascar. So we really don't know what they're going to do in terms of like how they integrate into food webs, because there's nothing that is exactly the same as them. So yeah, and then the second part of your question was, what do they do?
Yes, okay, just to follow up on that
Mhm.
technology aspect of this. Of course, like you think they're generally they eat everything, but are there studies in Africa that have looked at what they are actually eating? Because even amongst generalist species, you will find that they will have certain fish that is more than others.
Yeah, so
That's good.
yeah, that's a really interesting point, and something that I'm hoping to maybe answer with the ecomorphology stuff that I mentioned as well. Because like that certain elements of their morphology, they might be better at like eating certain things, even though they are generalists, like maybe some elements of their morphology make them prefer certain things. But yeah, that is a really interesting point.
And then, Gwen, please.
I know someone mentioned you were going to look at the guts.
Yes, so we can see what they're eating.
You're going to use.
Yes.
We're doing, however, just need to clarify that we're looking at gut microbiome, which will give us more of a kind of health.
So would it be worthwhile overlaying metabolic coding, since you've got the DNA extracted anyways?
Potentially, I think there are plans to do some of that, because we'll be doing that in collaboration with some colleagues in the US. We're trying to establish a protocol for that. The problem is that for a lot of invertebrates, for what Emma is doing, there is really no database for you to run your DNA. So that's the problem. So you rely on, when you're doing those kind of exploratory things, you rely on the database being present. And for invertebrates, they're famously absent or terrible. So it's difficult. But yes, that would be the.
Yeah, you can go ahead, sorry.
Thanks everyone for the talk. Maybe I missed this, but what feeds on crayfish? Like, what are the objectives, and did you ever consider biocontrol?
So, well, Julie's involved, and she's been talking about maybe developing an RNA virus for crayfish, which I think could work pretty well here, because there are not native crayfish, so be less risks in a context like Europe where there are native crayfish. That might be more risky, but I don't really know a huge amount about the biocontrol aspect. That's Julie's forte. And she also asked about what feeds on them. So fish will eat them when they're small. They'll eat each other as well if they can get their hands on each. We've had that a lot in our tanks. But yeah, I think it's mostly just fish. And yeah, otters will eat them as well. That's a good point. So yeah, things will naturally eat them, but the issue with them is that they won't eat them at a fast enough rate to sort of naturally control them, because the female crayfish can release like 100 offspring from one reproductive event. So yeah, you need a lot of otters to eat that many.
And with things like fish, when they get attacked, they go like this.
Yeah, that's.
And it's anything that's in, like, it makes it a lot harder to.
So yeah, you'd probably only be taking out the small crayfish and then left with like big ones.
Yeah, sorry, Albert. Yep, you can go ahead. Yeah, you can go ahead. Albert.
Yeah, thanks. Thanks, Emma. In terms of the impact on the invertebrates, because you're using SAS data, which is the identification is up to family level.
Yeah.
So I'm just wondering whether if you increase the resolution maybe to generic or species, the picture might potentially change in terms of the impact.
Now, I'm glad you mentioned that because that's something that I didn't include on my future work slide, but it's something that we're doing. So I have a friend called Bradley who works with Gordon O'Brien, who is he's got SAIAB data and has identified everything to species level. So we're going to do from Kruger, from the same sites that we did the 2025 data. So we're going to see what taxonomic resolution you need to sort of detect impacts. So we are looking into that, and yeah, watch this space.
And the functional diversity index would help to broaden that then.
Yeah. No, thank you. Yeah, but with that, I just need to do some stuff with the trait. There is a trait database for macroinvertebrates, but I'm doing some, it's called fuzzy coding to account for like variation in like life cycle. That they're quite flexible in what they do. So yeah.
How about the crabs?
What? Sorry.
How about interactions with the crabs?
That is
We're
something
working on it.
Danai's working on. as well.
And we're currently working on it, have a student working on it. So basically, because we did see and catch a lot of crabs in the same traps, every single crab that we've detected has been recorded with the same kind of morphology measurements. We've taken the same measurements from crabs and crayfish. And we are working on comparing the morphology between, and there is evidence that something is going on, but we haven't fully analyzed that data set yet. So,
and we also
work in
have
progress.
stable isotopes from the crabs as well, so we can look at the extent of trophic niche overlap between the crabs and the crayfish.
yes, we're very interested.
Answer that question.
In other systems with that, we've seen some overlap too.
Yeah.
Which is fine.
Try just opportunistically in the field of sort of a cage match sort of thing of a bucket.
People have done that. Josie did a study comparing at pinch strength of the crayfish and the crabs, and the crayfish had stronger claws, which is they might be better competitors. But yeah, that would be quite fun lab work. I think stuff has been done on that, but I can't remember off the top of my head the reference.
But we're working on
What's that? The chance
Ronella and Tumi has a question.
Thanks, Emma. I know your focus is mainly on macroinvertebrates, but have you observed any shifts on fish community?
The next talk will answer that question.
Next door. You'll get 3 minutes.
Cool. And then Tumi has a question.
Thank you, Emma, for the presentation. And mine is probably going to be linked to Albert's question, but in a different way. So I saw your presentation, I think it was Group 1, where you go to 2022, and then there was a shift. Then I realized that actually, I think that's when I started here. And then, but when I go back and then I look at what will be the impact of people sampling in these areas, because you look at the park, there's like some turnover in different scientists sampling these sites, and then you find that some are more efficient, they are well clued, and then some they are not. So what will be the impact of that and the sampling effort, obviously?
Yeah, that is a brilliant point, because obviously over the however many 10 years that they've set, I'm sure that the scientists going out to do the sampling has changed. All we can hope is that it is a standardized protocol that people will have followed, with the same amount of kick sampling being done. Obviously, maybe certain people might have higher kick strength than others, but yeah, there's unfortunately, unless we had when certain people did certain things and then we included that in the analysis as like a variable. But yeah, you can say it's a standardized protocol that people will have followed with the same amount of kicking, and yeah.
Cool. Okay, thank you very much, Emma. That was nice. Okay, and then last speaker, James. So what? The room is full of fish experts, so good luck. So James is also a PhD candidate But James is going to look at the impact also of crayfish, but also looking mainly on the impact of native freshwater fish communities. So yeah, over to you, James.
Yeah, so I don't really know how much time I've got, but I was going to also briefly go over what I'm doing. But basically, it's what Luba's just said. I'm looking at trying to work out how freshwater invasive species impacts vary across space and time, and how we can simply predict when and where impact will be greatest to inform management. And all of this I'm looking at through the lens of fish. That's me in the UK, obviously very different sampling to here in South Africa. So either the impact of invasive fish on native communities or an invasive species on native fish communities. So in the case of this, it is how are native fish communities affected by invasion. I was going to make a joke about how, you know, I had all of this time left to use up all the remaining time. It doesn't seem appropriate now, but that's fine. So just going through this very briefly, you've seen all the context. Introduced into the park in 2016, were since found in 2022 in the SAIAB, and they're polytrophic omnivores, which means they just feed on everything. And they also compete with macroinverts and with fish. At a fish level, this is what I'll be focusing on, they seem to eat eggs and juveniles, as well as outcompeting a wide range of them for their food sources. And like I said, I'll be focusing on fish. It's particularly concerning their impacts on fish because this is focused with what they do to fish catchers. Obviously, in the Kruger, there aren't any fisheries, but there are in sort of community lands and buffer zones. But the Kruger is also connected to 3 different countries. So we want to know what impact they're having and where it's the worst. So this project aimed. It's got three aims: what, why, and when. So what species are being affected? So we want to know how the communities vary between different sites, which I think was one of the questions asked earlier. And we predicted that a higher abundance of crayfish, we'd see different communities. And why? So why are these individuals being affected? So we looked at this using functional traits. So is it a particular functional group? Like when I'm talking about functional traits, I mean, what level of the water they're feeding in, or how do they reproduce? Do they scatter their eggs? Do they build nests, which they guard? Are they mouth brooders and things like that? And we predicted that that there will be less clutch tending, nesting, and guarding species, because there will be an increased sort of predatory pressure from the crayfish. And I thought that for egg scatterers, generally, you get a lot more eggs. So even if there is some predation pressure, it won't be felt as strongly as it would be by individuals that are having to guard eggs from these horrible little snippy fellows. So finally, when? So at what stage in the fish's life cycle are they being affected? Is it adult fish? Is it juveniles? Is it eggs? Although we just looked at juveniles versus adults. And I predicted that at a higher abundance, we'd see less recruitment of fish. So there'll be less younger fish. So to get into it very briefly, surveyed across 2 years, and the results presented are from multiple sites across the 3 rivers. We collected crayfish by trapping and fish through electric fishing. The data I'm presenting on crayfish abundance is just the trapping, because that was the standardized stuff, and the actual electro fishing I can't give catch per units on, because it was sort of, shit, we've seen a crayfish over here, let's go and try and catch it, which doesn't really work for standardized abundances. So moving on swiftly to the why. So this is the functional traits. We looked at sort of functional evenness, functional richness, functional dispersion, and functional divergence. So functional richness is just the number of different functional traits. Evenness is how they're distributed in terms of abundance. So if there's sort of empty niche space that can be invaded or not. Dispersion is how clumped traits are, or like whether we've got a very spread out range of different traits, or if they're all of a certain type. And divergence is whether the most abundant species have more extreme traits, so more specialized functions or less specialized functions. So this is a quick slide because we found no significant effect of crayfish abundance on any of the functional trait metrics. So there's no effect going on, which was a little bit strange, but as we go through the rest of the presentation, you can see how we can pull out some information about the functional ecology of fishes being affected based on imprints for other things. So there was no effect on crayfish at a functional level. That brings us swiftly on to what. So what species are being affected? And again, at a community level, we saw that there was no significant effect of crayfish on taxonomic community. It's interesting, so at a community level, we're seeing no impact of the crayfish. However, for taxonomy, if you looked at it on an individual species level, there were 2 fish that their presence or absence was significantly associated with the presence or absence of crayfish. So the elephant fish and the Tilapia sparmani. This one's my favourite, so this was a bit gutting to see. But basically, it looks like when crayfish are present, these fish are rare, and when crayfish are absent, they're much, much more common. And both these fish and the sites they were found in, there were sites where in 2025 they dropped off completely, and we didn't see them at all, where previously they've been pretty consistent, certainly in aggregated data sets up to that point. So there's some effect going on these, and these are both either clutch tending or guarding species. So both of these are species which are guarding eggs that have been laid. Then that brings us on to when. So at what stage in fish's life are they being affected? And what does that look like? So I'm looking at this using length frequency histograms. If you're not familiar with them, here is a mock-up of a length frequency. This is hypothetical data. And what you need to be looking at is comparing the invaded to the uninvaded. And if we see, this is what it would look like if there's no effect. So we see a very similar sort of size distribution, and the peaks here are roughly in the same location. If there is an effect, if you're looking here, we should expect to see a drop off in smaller sizes as younger fish are being predated. And it doesn't show as well with the density plot, but there'll probably be a shift of the peak slightly further to the right. But this is simulated data, so it's too perfect. So moving on to some real data. I'm going to start with OMOS. This is the Mozambique tilapia, and this is one where we see very little effect of crayfish. So you can see that in fact, generally, all of these size distributions are quite similar, and certainly in terms of smaller fish and the smaller size distributions, they all sort of have roughly the same amount here. There's slightly more effect in this invaded site, which is a little bit odd, but it's only small, so we'll not worry about it too much. But yeah, you see very little effect on the Oreochromis mozambicus. Moving on to one with slightly more effect, this is the red breast. And we can see that in the two uninvaded sites, we're lacking any size classes in this area. And the invaded site, we see it represented. Obviously, this is quite a small sample size on this one, but I had to make do with what we've got based on the data. And you can see that there's a representation of this smaller size class, which we didn't observe in these two other ones. You can also see sort of these two peaks a bit further to the right, and then we get this one here that we don't see for the other ones. Then finally, this is one of the Enteronius species, and we see a much more dramatic effect. So you can see that in the uninvaded site, we've got this really nice, well-represented spread at smaller size classes, and that's not represented at all in the Sand River, and is only represented a little bit in Antalistia. And what's interesting is that if you look at the abundance, there are 4 times more crayfish in the sand than there are in Antalistia. So we see this increased loss of smaller Enteronius at a site with more crayfish in, as opposed to those with less crayfish in. So essentially, to sort of summarize that section, we're seeing that a lot of these species are either egg scattering or guarding species, and we're seeing a lot of recruitment across a range of different species. There are other graphs. I didn't want to do 5,000, but you see it also in yellowfish and a load of other different Enteronius as well. So there's something happening to the recruitment of these species. But for other fish like the Oreochromis mozambicus or the southern mouth brooder, which is a hint, P-feed, Sucrinabis philander, we see that there's no effect at all. Both of these are mouth brooding species, which is really, really cool because we see no effect on these species which mostly protect their fry in their mouth, where the crayfish can't get to them or struggles get to them. We're seeing a lot more effect on unprotected fry or more vulnerable fry or eggs, which is pretty cool. Obviously not great for the unprotected ones, but interesting from an ecological perspective. So to bring it all together, we're currently seeing no effect at a community level. However, there are 2 species individually that seem particularly at risk, and these are both sort of guarding or clutch tending species. And we're seeing a lot of recruitment across a range of different species. And it suggests that the crayfish are having subtle effects on individual species, either through predation of their young or an increased sort of energy expenditure defending from crayfish. But there's nothing extremely dramatic happening yet, which sort of brings me on to the point of, if we act now, we can say we'll protect these rivers and certainly stop them from going further. Particularly as we're seeing with Emma, there's a macroinvert response, and that's generally what responds first, and then there'll be a lag before we start to see really dramatic community level change. So fortunately, we're not seeing that at the moment, which is a really, really good sign. But we are sort of seeing little telltales that it might come about in the end. So thank you for everyone that helped make this possible. It was a real pleasure. It was a pleasure to work with it.
We have questions for James. Question for Manja.
Hi, James. Thanks for a very interesting presentation.
My pleasure.
Some worrying results there. I see that you guys have done a lot of work inside the park, but a lot of those systems start outside the park.
Yeah.
The bigger systems. So I just wanted to know what is the state of crayfish invasion outside the park? Is there data or is there a gap that needs to be addressed?
Yeah.
Something.
It's probably a gap that needs to be addressed. So I'm going to look at these two here. Correct me if I'm wrong. There's some knowledge of we've got spotty patches where we know they've been seen, but in terms like there's some in Eswatini and there's some.
Yeah, so we know quite a bit about the spread in Zambia and Zimbabwe. That's kind of the immediate closer back, but we currently have no idea what's going on in Mozambique, for example.
And in terms of like immediate surrounding of the park, there's no data there. We're not aware of that. Yeah, definitely does. Yeah. So, question from the back.
Question from Roger.
Yeah, so your marketing that doesn't look like online system. I don't know what you think, Albert. That looks like very much.
Well, I might have put the wrong photo.
I think you need to look very often you get them together.
So that was not a photo that I took in the field because most of those got chucked back in. So great.
Very often you get them very patchily distributed. You get hundreds in one place and nothing in another place, and they're not in one crayfish.
Cool.
So that might mix up your results a bit.
And functionally, are they different?
Yeah. Totally different.
Okay, cool. Well, I mean, that photo wasn't from the field.
Yeah.
So I mean, in terms of our results, I'm hoping that it was people like Dummy and Lucas Wells helping. Dummy was passing the blame. Dummy was IDing them, so hopefully Dummy was IDing them correctly. But that's interesting. I might come and ask. Okay, perfect. Okay, cool.
Are there sections where there's also invasive fish and also there's overlap with crayfish? Or
we got like, I think there was one record of largemouth bass.
Okay.
And then Clarius as well is invasive in the Kruger. I know it's no, because I know it's native too. So, but like in terms of its native range, so that's fine. And yeah. So we had one record of a largemouth bass, but other than that, that's not.
And then awareness from you guys, are we doing enough or there's still lots to be done in terms of awareness campaigns? And then suggestions, how can we go about in terms of notifying or making people aware of this?
We've just started doing that. I think it's starting to get so. And you saw some of the infographics that Danai showing up. They're getting spread around. I think we're trying to kick off, like community groups and things as well.
Yeah, we've been to 2 workshops while we've been in South Africa, trying to address how we go about the management and how we trying to come up with like communication strategies for getting the message out there, because that's going to be the biggest thing, is making people, like a lot of people maybe don't know what a crayfish is. I didn't before I started my PhD, but yeah, that's the most important thing at the moment.
We're working on it. Policy briefs, infographics, raising awareness in general. It is a work in progress, and our experience even last week talking to people at the new site was that the moment we mentioned the fisheries, people were genuinely interested and asked for questions. And so hopefully.
And then communities around, how do they feel about catfish? Is it something they will eat, or is it
catfish or crayfish?
Crayfish. Is it a good pet in terms of colors? Or
it seems to be popular, is what we're learning. We actually managed to get a sample from a pet shop, and we will be testing the genetics of it. So that was pop up around the area. But that's a different species. That's from the red swan. The Redclaw has been introduced exclusively for aquaculture, as far as I'm aware. The red swamp, that's smaller and cuter and prettier and comes in fancy colors, seems to be a pet.
Like you said, there's a bit of a gap in terms of what's going on around the park and like distribution, and maybe it would be interesting to see. I know there's work going on in other places about how people feel about crayfish. We've touched on that, and often they're approached with, then they're not liked very much because then people aren't used to them as a food source. So I'm reluctant to use them.
Okay, there's a question from Dumi.
Thanks, Luba. Actually, it's not a question. I just got cut off. So I just want to talk about the awareness and how people are feeling around the park. So to address that component, we have a project now that is going to be done by Dileri. She's going to look at the socio-ecological aspect of crayfish. So we have some sites around the park, the neighboring communities where we'll go and try to understand what are their views, and we'll also try and sample around the communities because the missing link for us in the park is that we are unable to do the surrounding communities. That's why we going to do that with researchers. I know Tatenda and his team have done some sampling outside the park, going in the Crocodile River, going all the way up to Nelspruit, and we waiting for the results and to see what is the distribution and spread. So it is mostly around working with the communities and see what is their view and how can we stimulate the interactions. And the other thing, having the biggest problem with the park is these are transboundary systems where the management is not only dependent on the park, but it's more on collaboration where you are dealing with different countries. That's why we had like this workshop where we had Limcom, which is covering Mozambique, Zimbabwe, and Botswana, and then Inmacom, which covers Eswatini and South Africa and Mozambique, so that we can try to harmonize the policies and how can we manage, because at the end of the day, we as a park cannot do it alone. We depend on different stakeholders to contribute to the management. We know that eradicating the species will be a huge challenge. That is why we are trying to get around that. And also the number helps us in that regard. So yeah, that's what I wanted to say. Unless if there are more questions.
Cool. And then collaborations, applying for funding, is that possible? How can we chat with you guys more and then share resources?
Yeah, it's definitely possible. That's partly what's been going on over the last 2 weeks, is trying to reach out and work with like DFFE as well as NIMCON and INNICON and things like that. In terms of sharing resources, I think that'd be having a think and working out what we can do from that. I mean, Josie probably had many ideas already, but yeah.
Cool. Thanks, everyone. Oh, sorry. Understand.
How detrimental are these from the native areas?
It's funny, isn't it?
They're having a huge impact here as invasives, but how detrimental are they?
I suppose in their native areas, they've been there for however long. So anything that is there is stuff that can survive being there without being like overpredated or things like that. Unless there's
natural predators to
they've got quite a lot of natural predators that will just opportunistically like take them. So like we've said, some fish will eat them, otters like them. There are some birds that will eat them as well. In the native areas as well, yeah.
There's nothing different there than here.
Not necessarily. It's something I'm working on at the moment with the crayfish database. I'm particularly interested from an evolutionary ecology perspective, comparing native versus invasive range for cases where I do have data for the exact same species. But it's the reason why we often include evolutionary novelty in the definition of an invasive.
What would be interesting though would be looking at whether in established native or established invasive populations, whether they reach in similar systems, whether they reach the same abundance levels. Because that would be, it would be cool to see if actually you get very similar densities in per whatever meter squared, or if they're actually particularly high in invasive systems because there's some control that's not being carried across that.
The environment plays a role as well. I've seen that from work on invasives from previous work that I did my PhD, for example, and there is evidence that they might just be there and then just their impact becomes much higher when the environment starts resembling their native environment. It's why I did for my PhD, for example. So I know it can happen. So it could be the case with the crayfish as well.
Okay, we have to go back to work. So thanks to the guys online. Yeah, thank you.